Your first question, and the question is the fixative most commonly used in TEM is. TEM is nothing but transition electron microscopy. TEM is widely used to study the internal structure of the cell, especially the cell organelles. You know that SEM is scanning electron microscopy. It is used to study the external structure of the cell. Okay, so in TEM, the fixative user is the aldehyde derivative. So when you are looking at this particular question, you can see that there are three aldehydes mentioned in the option. They are acetaldehyde, glyceraldehyde and glutaraldehyde. But the most appropriate answer is the glutaraldehyde. Then how this glutaraldehyde act as a fixative? This glutaraldehyde react with the amino group of basic amino acids such as arginine and lysine and produces cross-linked protein. Okay, this cross-linkage maintains the proper fixation. So, the correct option is the D, that is the glutaraldehyde. Here is your second question. Presbycusis is. It's a condition as a person gets older and older, you can see hearing impairment, loss of hearing abilities. That condition is known as presbycusis. Okay, so when you are analyzing the four options, you can see that the correct answer is the fourth option, that is hearing loss due to old age. As a person gets older and older, we can see hearing impairment, loss of hearing abilities gradually, and that condition is known as presbycusis. Okay, so the correct option is D. The answer is D. Let us discuss the next question. The standard deviation if then the options are root mean square deviation, mean square deviation, square of variance, square of mean square deviation. To find out the standard deviation, at first you have to work out the mean, that is simple average of those numbers given. Then subtract the mean and square the result. Then work out the mean of the square difference, then take the square root. Okay, then we will get the actual standard deviation. For calculating standard deviation, we find out the mean, that is the average of the value. Then we squared the value and finally we took the root of the value. So the correct answer for this question is option A, root mean square deviation. Okay. The next question is the mineral deposit that is transported to a site by wind is called. Okay, now we can check the options. First option is colluvial deposit. Colluvial deposits are those deposits usually deposited by the action of gravity. Okay, and the option B is aeolian deposit. These are the actual deposit which are get deposited by the activity of wind. So the correct answer is B. Okay, the option C is alluvial deposit. These are the sediments which are deposited by the action of a river. Okay, and the final option glacial deposit, they get deposited by the activity of moving glacier, that is large icebergs, isn't it? Okay, so for this question the correct option is the option B, that is the aeolian deposit. And these deposits are get deposited by the activity of wind. The fifth question is occurrence of a single act of reproduction during the lifetime of an organism is called. Okay, based on the active reproductive period or active reproductive episodes, the organism can be classified into two types, and they are simulparity and heteroparity. Simulparity means those organisms having a single reproductive episode during the entire life cycle. Okay, whereas heteroparity means organisms are having multiple reproductive episodes in their entire life cycle. So, simulparity and heteroparity are those zoological terms. When we come to botany, the term which is similar to simulparity is what? Monocarpic. The plants which are having the capacity to produce seeds and fruit only one time within the entire life cycle. Okay, and the term of botany related to heteroparity is polycarpic. Those plants having the capacity to produce fruits and seeds multiple times, several times in their entire life cycle. Okay, so for this question, the correct option is C, that is similarity. The immunoglobulin seen in protein with X-linked agammoglobulinemia is, the options are IgD, IgM, IgG, IgA. Okay, let us understand what is agammoglobulinemia. X-linked agammoglobulinemia is a recessive genetic disorder. This disorder is mainly due to the presence of a recessive gene in X chromosome. You know that X chromosome is one of the sex chromosome. Okay, and in normal person, in X chromosome there is a dominant gene, and that dominant gene will code for an enzyme known as tyrosine kinase, and this tyrosine kinase enzyme is essential for the maturation of B lymphocytes and which will enable proper immunity. But those patients having this disease, that is agammoglobulinemia, lack the dominant gene. Instead of dominant gene, they possess a recessive gene, and that recessive gene is unable to produce the enzyme tyrosine kinase. Hence, in those patients, the immature B lymphocyte never matured and lead to a condition called X-linked agammoglobulinemia. Okay, and these patients are severely susceptible to various type of bacterial infection, and the major bacteria which affect those patients include Streptococcus, Staphylococcus, and Haemophilus. Okay, so let us once again look at the question. The question is, the only immunoglobulin seen in patient with X-linked agammoglobulinemia. Okay, you know that in agammoglobulinemia, those patients are unable to produce antibodies, isn't it? Because they lack the B lymphocyte production, isn't it? But those patients having this condition is capable of producing an immunoglobulin, namely IgG. Okay, immunoglobulin G. So the correct answer for this question is option C, IgG. Remember, those patients having X-linked agammoglobulinemia is having the capacity to produce what? IgG. So the correct answer is option C. You know that the immunoglobulins are globular shaped protein capable of providing immunity by destructing pathogens, including antigens. Okay. And the major immunoglobulins are IgG, IgA, IgM, IgD, and IgE. Okay, in order to remember these immunoglobulin, we have to remember a code that is GAMDE, G-A-M-D-E. Okay, G for IgG, A for IgA, M for IgM, and D for IgD, and E for IgE. Okay, the code is GAMDE. Okay, of which the largest immunoglobulin in structure is IgM. IgM is a pentameric structure. Okay. Okay, the next question is, the fishes migrating within fresh water only are called. The options are Potamodromus, Anadromus, Diadromus, and the final option is Anatropus. Potamodromous fishes are migratory fishes whose migration is restricted to freshwater only. Diadromous fishes are truly migratory fishes. They can migrate between sea and freshwater. Anadromous fishes are a type of diadromous fishes. You know that diadromous fishes are capable of migrating between sea and freshwater. So anadromous fishes are a type of diadromous fishes which spend a major part of their life in sea, but they migrate to freshwater only at the time of egg laying, that is spawning. You know that spawning is a term which means the release or deposition of egg by fishes, certain mollusks, like that. Okay. In this question, the fourth option is Anatropus. Anatropus is usually a botanical term related to ovule. That is, 180-degree rotated ovules are commonly known as Anatropous ovule. In the case of angiosperm, The majority of the angiosperm possess this type of ovule, namely anatropous ovule. Remember, ovule is also considered as the megasporangium. Okay, within the ovule, you can see the female gametophyte of angiosperm, namely embryo sac. But in the case of gymnosperm, the female gametophyte is not the embryo sac, it is the endosperm. Okay, so the correct option for this question is option A, Potamodromus. The eighth question is World Conservation Days. Okay, the answer given by PSC is December 3. But December 3 actually is a day for handicapped and not for the World Conservation Day. Actually, the correct answer for this question is July 28th. And the World Wildlife Conservation Day is celebrated on December 4. Okay, I think this question was deleted at that time. Let's go through the next question. And the question is, hill station like Munnar, Ponmudi and Wayanad belong to dash biogeographic zone. You know that all these regions, mainly Munnar, Ponmudi and Wayanad belongs to our Western Ghats. Okay, so the correct answer is the option D, Western Ghats. It's one of the biodiversity rich region. Okay, the 10th question is, the basa of fabricus is a lymphoid organ in. The options are amphibian, reptile, bird, mammal. From this slide, it is very clear that basa of fabricus is a structure related to birds. It is actually a pouch found on the dorsal part of the cloaca of birds. During day 15, it becomes a lymphoid organ and it becomes fully functional near hatching. Okay, 7 to 13 weeks of puberty or attaining the age of 7 to 13 weeks, it get involuted and it is a immunocompetent structure capable of producing lymphocytes or B cells that are known as basal lymphocytes. Okay, then migrate to spleen. So, correct option for this question is option C, bird. Let's begin our discussion. Okay, the question is, the common mark and recapture method used to estimate the total population density in a defined area is. Okay, before going to the answer, at first we can make a look at all the options. The option A, Lotka-Volterra index, option B, Lincoln index, option C, Liebig index, and option D, Haber index. Okay, at first we can briefly understand all these options. Okay, Lotka-Volterra model or Lotka-Volterra index was first proposed or developed by Lotka and Volterra to explain prey-predator relationship. So, remember, Lotka-Volterra model is associated with the prey-predator relationship. They are actually explaining by this model the prey-predator relationship. Okay. The next one is the Lincoln index. It is widely used to estimate animal population size in a particular area. It is very difficult to count each and every animal present in a particular area to estimate population size. So, what is actually doing in this method is, at first we are capturing that particular animal, we want to know their population density, then mark them with a tag, then release them and wait for a particular period of time, then recapture these animals again, and by applying a suitable formula we are calculating the approximate population density of that particular animal in that particular area. Okay, as far as your CSIR net license examination is concerned, this method is of immense significance. You people will get at least one problem from this portion. Okay, immediately I will upload a video regarding this method and the easy method to find out that solution for that particular problem. Next one is the Liebig's index, it is associated with a law known as Liebig's law. Liebig's law is one of the laws explaining law of limiting factor. We already studied about the Blackman's law of limiting factor in photosynthesis. Okay. What do you mean by Blackman's law of limiting factor? When a chemical process or a biological process is controlled by several factors and the rate or pace of that particular process is determined by the factor which is nearest to the minimal value, and it is known as the Blackman's law of limiting factor. Okay, in that sense, it is very similar to Blackman's law of limiting factor. Liebig's law states that as far as the population is concerned, the growth of the population is determined by the limiting factor, that is the factor which is nearest to the minimal value, and not the abundance factor, the most available factor. Okay, this is the Liebig's law. For example, we can consider the growth of population. Okay, the growth of population is determined by the availability of nutrients, the availability of oxygen, and the climatic factors. Okay, if there is a lot of nutrients are available and abundant amount of oxygen is available, but the climatic condition, when it is limited, then the growth and development of the population will not take place properly. Okay, because the growth and development of that particular population is influenced by that lowest factor or limiting factor, namely climatic factor. Okay, this Liebig's law or index means. Look at the fourth option. It is Haber index. Haber index is related to Haber process. It is an artificial nitrogen fixing process in which the nitrogen is fixed in the form of ammonia. It is also known as Haber-Bosch process. Okay, so from this option, now you know that the correct answer is what? The correct answer is the Lincoln index. This index is widely used to estimate the population density in a particular area by recapture method, the mark and recapture method. Okay, I am inviting your attention to the next question. The question is, the chi-square is 0 when the option A is expected frequency is lesser than the observed frequency. Option B is expected frequency is equal to the observed frequency. Option C is expected frequency is greater than the observed frequency. And the final option is expected frequency is double that of observed frequency. Okay, at first we can understand what is chi-square test in brief. Chi-square test is one of the important tests to find out the significance. Okay, it was developed by Carl Pearson in 1900. Okay, and the chi-square test is a non-parametric test, and it is not based on any assumption or distribution of any variable. And these statistical tests mainly follow specific distribution known as chi-square distribution. Okay, in short, the test we use to measure the difference between what is observed and what is expected, according to an assumed hypothesis called as chi-square test. Okay, so this chi-square test is widely used to find out the significance based on the expected and observed value. This is the formula for the calculation of chi-square. Chi-square is usually designated by a symbol x square, and the equation is chi-square is equal to sigma O minus E the whole square divided by E, where O stands for observed frequency and E for expected frequency. Okay, look at the third statement. If two distributions, that is observed and theoretical, are exactly alike, the chi-square is equal to 0. Okay, now I think you people got the answer. The correct answer for this question is option B, that is expected frequency is equal to the observed frequency. The next question is a taxon based on fossilized work including trails, tracks and burrows by an animal is. Option A is hypotype, option B is hyponym, option C is ichnotaxon, and the final option is idiotype. Look at the first option, that is hypotype. Hypotype is a type specimen which is used to describe the plants and animal. Okay, and in hypotype, a particular plant or animal is described based on the data available on previous publication, or it is described based on certain figures. Such a type of type specimen is known as hypotype. Option B is hypodigm. Hypodigm means it's a sample from which the characters of a particular plant or animal or a population is being described. A taxon based on fossilized work is known as ichnotaxon. That is the option C. Idiotype is a type specimen explained by an author, but it is collected from a region other than the region described by the author. So the correct option for this question is option C, ichnotaxon. Okay, the next question is, the neutralist theory of evolution was proposed by option A, Sacco, option B, Garrow, option C, Moto Kimura, option D, Andreas Wagner. The neutral theory or neutralistic theory of molecular evolution was developed and advanced by the scientist Moto Kimura. And according to this theory, there are two basic principles. One is the most natural population harbor a high level of genetic variation, and this high level of genetic variation is much more than the expected, which is due to natural selection. And many mutations in sequence of genes do not alter the proteins encoded by those genes, because those mutation might be nonsense mutation or missense mutation like that. So the answer for this question is option C, Moto Kimura. The next question is, the graph obtained by plotting reciprocal of initial velocity and substrate concentration is option A, E.D. Hofstee plot, B. Lineweaver plot, C. J plot, and D. Ramachandran plot. Okay, this question is from the portion that is from the topic enzyme kinetics. This is one of the important equation in the topic enzyme kinetics. The equation is known as Michaelis-Menten equation. The equation is that V0 is equal to Vmax into S divided by Km plus S. Here V0 is the initial velocity, Vmax is the maximum velocity, and the S is the substrate concentration, and Km is the Michaelis-Menten constant. Km is actually the concentration required to produce half the maximum velocity. That is, Km is the concentration, substrate concentration required to produce half the maximum velocity. This is a basic Michaelis-Menten equation. There are certain drawbacks for the Michaelis-Menten equation. For our easiness, we are taking the reciprocal of Michaelis-Menten equation. Okay, you know that the Michaelis-Menten equation is V0 is equal to Vmax into S divided by Km plus S. When we are taking the reciprocal of that Michaelis-Menten equation, it will be 1 by V0 is equal to Km divided by Vmax into S plus 1 by Vmax. Okay, when we plot this equation on a graph, then we will get a graph like that. Okay, and this graph is known as Lineweaver-Burke plot. Okay, where y is equal to mx. It is straight line equation. It is the equation to find out what? Slope. Okay, this is actually the Lineweaver-Burke plot, and this Lineweaver-Burke plot is obtained by plotting reciprocal of initial velocity and substrate concentration, isn't it? So the correct option for this question is option B, Lineweaver-Burke plot. It is simply the reciprocal plot of Michaelis-Menten equation. Okay, and this Lineweaver-Burke plot as well as this Michaelis-Menten equation is of much importance as your CSR Net Life Science exam is concerned. Okay, look at this question. The question is, Zellweger syndrome is due to option A, inefficient peroximal beta oxidation, option B, inefficient mitochondrial beta oxidation, option C, inefficient glycosomal beta oxidation, and the last option is inefficient omega oxidation. If you are analyzing the options of the question provided, you can see a term beta oxidation. Okay, here beta oxidation means beta oxidation of fatty acid. Before going to beta oxidation, we can check what is fatty acid. You know that fatty acids are the building blocks of fats or lipid. They are organic acids having carboxyl group at its one end. Okay, and the carbon atom which is present near to the carboxyl group is known as alpha carbon, and the carbon atom which is present just adjacent to the alpha carbon atom is known as beta carbon atom, and the last carbon atom is known as the omega carbon. Okay, omega carbon is usually represented by methyl group in a very fatty acid. So, beta oxidation of fatty acid is a breakdown of fatty acid into two carbon molecule, namely acetyl coenzyme A. Suppose if we are going to break down a 16 carbon containing fatty acid, namely palmitic acid, then what will be the product we obtained? The product will be 2 carbon acetyl CoA and remaining 14 carbon fatty acyl CoA. Okay. Beta oxidation of fatty acid mainly occur in mitochondrial matrix. But in addition to mitochondrial matrix, it can also take place in the peroxisome and glyoxysome as well. What are glyoxysomes? Glyoxysomes are specialized peroxisome. They are found in plants having fatty or oily seeds. Okay, for the proper seed germination, this fat or oil has to be converted into sugar or carbohydrate, isn't it? Okay, and that process takes place by means of beta oxidation of fatty acid as well as by another cycle known as glycosylate cycle. Okay, and the key enzymes involved in glycosylate cycle is isocitrate lyase and malate synthase. I think the question about this key enzyme asked in the previous year's CSR net exam. So these two enzymes are very important. Okay, glycosomes are found in contact with the lipid bodies in cartilagens or endosperm where fatty acids are being converted into carbohydrate during germination. Remember, this glycosylate cycle can be seen in those seeds storing fat or oil. Okay, what is the role of this glycosylate cycle? To mobilize this oil or fat into soluble sugar, which will help for the proper germination of the seed. Okay. Then what is Zellweger syndrome? Zellweger syndrome is a peroxisome biogenesis disorder. The patient having this syndrome is provided with a very low amount of peroxisome. Okay, as a result, the beta oxidation of fatty acid will not properly take place in those patients. Okay, when we are analyzing their cell, we can see long chain fatty acid remaining as such. Why? Because the beta oxidation of fatty acid is taking place in very low rate. Okay, so the Zellweger syndrome is associated with the inefficiency of beta oxidation of fatty acid in peroxisome. Once again, I am inviting your attention to this question. Okay, so the Zellweger syndrome is due to, now you know that it is due to the inefficient peroximal beta oxidation. So the correct answer for this question is the option A. Okay. Okay, next question is name of a species or subspecies in which second or third component of the name repeat the generic name is called. Option A, chrysonym, B, synonym, C, homonym, and the last option is tautonym. The first option is chrysonym. What do you mean by chrysonym? Chrysonym is a cited use of a taxon name, usually a species name within a publication. In botanical and zoological nomenclature, synonyms are the different scientific names of a same taxon. For example, two names for the same species. But a homonym is a name for a taxon that is identical in spelling to another such a name, but that belongs to different taxon. So remember, synonym is the different name of a same taxon, whereas this homonym is identical in spelling but belongs to different taxon. Okay, what do you mean by tautonym? Tautonymy is a binomial in which both genus and species names are identical. That is, genus and specific epithets are identical. Okay, for example, Helianthus, then Sisyphus. Okay, but these tautonyms are not permitted by International Court for Nomenclature. Hence, it is said to be illegitimate. Okay. So, the correct answer for this question is option D, tautonym, where both the genus and species name are identical. Okay, the next question is the matrilineal most recent common ancestor is known as option A, Y chromosomal Eve, option B, historic Eve, option C, X chromosomal Eve, and option D, mitochondrial Eve. In the field of human genetics, the name mitochondrial Eve refers to the matrilineal most recent common ancestor of all currently living anatomically modern humans, who is estimated to have lived approximately 100,000 to 200,000 years ago. This is the most recent woman from whom all living humans today descend. Okay, and this mitochondrial Eve, that is the matrilineal most recent common ancestor, is abbreviatedly known as MRCA. So the correct answer for this question is option D, mitochondrial Eve. Next question is myoglobin is a protein existing in dash structure. The options are A, quaternary, B, tertiary, C, secondary, and D, primary. Okay, what are proteins? Proteins are the polymers of amino acids. Okay, the amino acids join together to form what? Protein. Okay, you know that all proteins are heteropolymer. That is, all proteins are made up of different type of amino acid. You can't see a protein with the same amino acid. Okay, and in protein you can see the four level of structural organization. They are known as primary structure, secondary structure, tertiary structure, and quaternary structure. In primary structure, the proteins are arranged in the form of a linear structure, where amino acids are arranged, held together by peptide bond, just like beads on string, isn't it? Okay, and this peptide bond is having the partial double bond character, even though it appears as a single bond, but it is having partial double bond character due to resonance effect. Okay, and from the primary structure, the orientation of amino acid arrangement and the position information of amino acid can be interfered. That is, which is the first amino acid, which is second amino acid, and which is the last amino acid, like that. Okay, and what is about secondary structure? When these proteins in the primary structure undergo folding or coiling or twisting, it will give rise to another structure known as secondary structure. You know that secondary structure is of two types, alpha helical structure and beta pleated sheet structure. Okay, then the tertiary structure. What is tertiary structure? When these proteins in the secondary structure arranged in the form of a woolen ball, okay, it will get a three-dimensional structure. It is known as tertiary structure. Tertiary structure is also known as three-dimensional structure. Okay, in order to get certain activity, the protein should be in tertiary structure. Okay, then what is quaternary structure? In quaternary structure, more than one polypeptide chains are involved. Okay, whereas in primary, secondary, and tertiary structure, only one polypeptide chain is involved. Okay, and these polypeptide chain together form a plate-like structure, or a rectangular shaped structure, or a spherical shaped structure, like that. Okay, such a type of structure is said to be quaternary structure. A typical example of protein occupy in quaternary structure is our hemoglobin. Okay, now let us look at the difference between the hemoglobin and myoglobin. Okay, hemoglobin is a red protein which is responsible for transporting oxygen in the blood of vertebrates, whereas myoglobin is a red protein with heme which carries and stores oxygen in the muscle. The molecular weight of hemoglobin is 64 kilodalton. For that of myoglobin is 16.7 kilodalton. And hemoglobin is composed of 4 polypeptide chain, whereas the myoglobin is composed of single polypeptide chain. Okay, remember that. And in the case of hemoglobin, it is a tetramer composed of 2 alpha and 2 beta units, whereas in myoglobin, it is a monomer and it lacks quaternary structure, but the quaternary structure is present in hemoglobin. Okay. And hemoglobin binds with 4 oxygen molecule at a time, whereas the myoglobin binds with a single oxygen molecule. Hence, myoglobin is a monomer and is made up of a single polypeptide chain, and its highest level of protein structure is tertiary. So, the structural organization of the protein myoglobin is tertiary structure. So the answer for this question is option D, primary. Remember, myoglobin is a protein made up of a single polypeptide chain and is having tertiary structure. Okay, next question is the sharing of ancestral characters by cells, which is called option A, synapomorphy, B, symplisomorphy, C, ontogeny, and the last option is phylogeny. Okay, let us discuss some important difference between synapomorphy and symplisomorphy. Okay, synapomorphy means a characteristic present in an ancestral species and shared exclusively by its evolutionary descendants. That is in a more or less modified form. Whereas symplisomorphy means what? An ancestral character shared by 2 or more taxa. That's most important difference. In synapomorphy, it describes derived character, whereas symplisomorphy describes an ancestral character. In synapomorphy, the character is shared by the most recent common ancestor. But in symplisomorphy, the character is shared by the earliest common ancestor. Example for synapomorphy is the presence of the same number of vertebrae in the necks of the mammals and giraffes. And the example for symplisomorphy is the presence of a vertebral column in the mammals and other vertebrates. Let us look at the major differences between ontogeny and phylogeny. Okay, in ontogeny, it deals with the development or developmental history of an individual organism, whereas in phylogeny, it deals with the study of relationship among different groups of organisms and their evolutionary development. Ontogeny gives the development history of an organism within its own lifetime, whereas phylogeny gives the evolutionary history of a species. Okay, ontogeny describes how a chicken came to life starting from a single cell, whereas phylogeny describes the evolutionary process of Gallus gallus, a particular species. Okay, and that particular species may include a lot of individuals, isn't it? Hence, ontogeny, the focus of study is an individual organism, whereas in phylogeny, okay, the relationship between different individuals considered at a time. Okay, now you people know the answer for this question, that is option B, symplisomorphy. Okay, this synapomorphy and symplisomorphy is an important topic when your CSIR net exam is concerned. Okay, the answer for this question is option B, symplisomorphy. Look at the question. The new era about to be added as formal unit of geological timescale is option A, Sapiocene, B, Homocene, C, Psychocene, and D, Anthropocene. What do you mean by geological timescale? The geological timescale is a record of the life forms and geological events in the Earth history. And scientists developed this timescale by studying the rock layers and fossils worldwide using radioactive dating technique. In geological timescale, the largest sections are called eons. Eons are subdivided into eras, and eras are further divided into periods, and periods are finally divided into epochs. Anthropocene is the current geological age, viewed as the period during which the human activity has been the dominant influence on climate and the environment. And it is a new era about to be added as a formal unit of geological timescale. Hence, the answer for this question is the option D, Anthropocene. The detailed video on geological timescale from the examination point will be uploaded later. Let's discuss the next question. The question is, which of the following is a protein structural motif involved in protein-protein binding? Option A, zinc finger motif. Option B, helix turn helix motif. Option C, leucine zipper motif. And the option D, hairpin motif. Let us understand motif in brief. What motif means? The protein from the same family is having certain conserved region, and these conserved region acting as a binding site or active site. Okay. What do you mean by conserved region? Consider there are 20 proteins, and these 20 proteins shared certain common region. Okay, and these common regions are considered as the conserved region or conserved sequences. Okay, hence the motif is nothing but the conserved region of protein of same family. Okay, and this motif acts as a binding site or active site to which a particular molecule binds. Okay, that molecule may be another protein or maybe DNA or RNA like that. Okay, however, these motifs are not usually detectable by sequence search techniques such as BLAST or FASTA. Okay, what do you mean by BLAST? BLAST? BLAST is Basic Local Alignment Search Tool. The full form of FASTA is Fast All. Fast All. Okay. In our question, the first option is zinc finger motif. What is zinc finger motif? It is a three-dimensional structure motif of the protein, characterized by the coordination of one or more zinc ion. Okay, when you are analyzing zinc finger motif, you can see the presence of zinc ion. And the zinc ion is held in place by two cysteine and two histidine R group. Okay, you know that cysteine is a sulfur-containing amino acid and histidine is a basic amino acid. And this zinc ion is held by two amino acids, namely cysteine and histidine. Okay, and they are present in the tandem repeats. Okay, they are present in tandem repeats. What do you mean by tandem repeats? When certain sequences are repeated in a chain-like manner, it is known as tandem repeats. Okay, and zinc finger motif is usually associated with the DNA and RNA. Or simply we can say that zinc finger motif is a region where DNA and RNA binds. Okay, so zinc finger motif is mainly meant for the binding of either DNA or RNA. Okay, the zinc ion holding the structure together. And the structure of zinc finger motif is maintained by whom? Maintained by simply by zinc ion. Okay, each zinc motif consists of 30 amino acids and folds into beta alpha structure. Okay, each zinc motif consisting of 30 amino acid and they have a conformation beta alpha structure. Okay, each finger primarily binds to a triplet within the DNA substrate. Okay, so zinc finger motif is mainly meant to bind with either DNA or with RNA and not with another protein. The B option in the question is helix turn helix motif. Okay, helix turn helix motif is a major structural motif capable of binding DNA. It is found in many regulatory proteins. It binds to the major group of DNA through a series of hydrogen bond and also through a weak bond, namely van der Waals interaction. And this bond is made with the basis of DNA. Okay, then the other alpha helix do not have any recognition property, but it stabilizes the interaction between protein and DNA. Okay, the recognition helix and its preceding helix always have same relative orientation. Hence the name helix turn helix motif. The option C in the question is leucine zipper motif. Leucine zipper is a common three-dimensional structural motif in protein. It is abbreviatedly known as ZIPs. Okay, and these zip-like motifs have been found in protein other than transcription factors. They are thought to be one of the general protein modules for protein-protein interaction. So, leucine zipper motif is mainly involved in protein-protein interaction. The D option in the question is hairpin motif. It is also known as beta hairpin. Okay, it is formed by two beta sheets that just look like what a hairpin like structure, hence the name hairpin motif. And hairpin motif is the actual site for protein folding. Okay, look at the question once again. Now you people know the correct answer for this question. The correct answer for the question is option C, leucine zipper motif. It is mainly involved in protein-protein binding. Okay, whereas zinc finger motif is involved in binding with either DNA or RNA. Helix turn helix motif is involved in the binding with DNA, especially with what? Major group of DNA. Whereas hairpin motif is involved in what? Protein folding. Okay. So the answer for this question is option C, leucine zipper motif. Let's go through the next question. The next question is Philadelphia chromosome is an example of chromosome where option A, segment is translocated. Option B, a segment is inverted. Option C, a segment is duplicated. Option D, a segment is looped. It is a question based on the proportion chromosomal aberration. What do you mean by chromosomal aberration? Chromosomal aberration is a type of chromosomal mutation which is due to change in the structure of chromosome. When a chromosomal mutation is due to change in the structure of chromosome, it is known as chromosomal aberration. It is of the following type. First one is the addition. When a segment of chromosome is added to an original chromosome, it is known as addition. When a segment of chromosomal fragment become deleted from the original chromosome, it is known as deletion. When a segment of original chromosome is get duplicated, it is known as duplication. When a segment of original chromosome get detached from the original chromosome and attached to the original chromosome in an inverted manner, 180-degree rotated manner, it is known as inversion. Okay, inversion can induce what? Positional effect. Remember, positional effect. Okay, and this inversion play an important role in gene regulation as well. Okay, then exchange of chromosomal fragment between two non-homologous chromosomes can be called as what? Translocation. Okay, for example, if there are two different chromosomes, we can call that those different chromosomes as non-homologous chromosome. When a particular chromosomal fragment from one chromosome is added to the another chromosome, it can be called as translocation. Okay, translocation is the exchange of chromosomal fragment between two non-homologous chromosomes, not homologous chromosome. When it comes to the homologous chromosome, it can be called as what? Linkage. It's a natural phenomena. Okay, linkage, which can be seen during what? Meiosis. You know that. Okay. So, there is one important point regarding this translocation is translocation is the exchange of chromosomal fragment between two linkage group. Between two linkage group. Whereas the crossing over or recombination is the exchange of chromosomal fragment within the linkage group. That is a difference. When chromosome get a loop-like or coil-like structure, then we can call that chromosome is having its one segment looped. What do you mean by Philadelphia chromosome? Philadelphia chromosome is a shortened chromosome 22 in human. Okay, the Philadelphia chromosome is abbreviatedly known as PH chromosome, which is due to the reciprocal translocation between the chromosome 9 and 22. And this reciprocal translocation leads to the production of a hybrid gene, and that hybrid gene will result in the increased production of an enzyme known as tyrosine kinase, and which eventually leads to a disease condition known as leukemia. That leukemia is especially known as chronic myeloid leukemia. Okay, so the Philadelphia chromosome is due to the reciprocal translocation between chromosome 9 and 22. Consider two chromosomes, chromosome A and chromosome B. When a fragment of chromosome B get detached from the chromosome B and attached to chromosome A, it is known as normal translocation or simple translocation. When both the A and B chromosome mutually exchange the chromosomal fragment, such a type of translocation is called what? Reciprocal translocation. Okay, in that Philadelphia chromosome, we can see the mutual exchange of chromosomal fragments between the long arm of chromosome 9 and 22. As a result, the length of the arm of chromosome 22 becomes shortened. That shortened chromosome number 22 is known as actually what? Philadelphia chromosome. And that Philadelphia chromosome leads to a disease condition known as chronic myeloid leukemia. Okay, once again I invite your attention to this question. Now you know the answer. The Philadelphia chromosome is an example of a chromosome where a segment is translocated. Philadelphia chromosome is a typical example for what? Reciprocal translocation. Okay, this translocation and recombination, when we are analyzing this translocation and recombination, we can see similarity, because in both the cases we can see the exchange of chromosomal fragments, isn't it? Then how they differ? In recombination, the exchange of chromosome fragment takes place between homologous chromosome, whereas in translocation, especially in reciprocal translocation, the exchange of chromosomal fragments takes place between two non-homologous chromosomes. Okay, homologous chromosomes are considered as the same linkage group, whereas non-homologous chromosomes are considered as not same linkage group. Hence, recombination takes place within the same linkage group, whereas this reciprocal translocation is not taking place within the same linkage group. Reciprocal translocation takes place between two different linkage groups. Remember, that's very important. Okay, so the correct answer for this question is option A. Next question is, a small molecule that by itself is incapable of eliciting an antibody response but can act as an epitope is called. The options are A. haptin, B. icosome, C. isotype, and D. integrin. The question, you can see a term epitope. What do you mean by epitope? Epitope is a region of antigen where antibody binds. That is, antibody binding site of antigen can be called as epitope. Okay, paratope is a term related to epitope. What is paratope? Paratope is a region of antibody where antigen binds. Okay, so epitope is present within antigen, whereas the paratope is present on antibody. Okay, epitope is the binding site present in antigen, whereas the paratope is the binding site present in antibody. Okay, you know that antigen is any foreign substance which enter into the body. Epitope can be also defined as antigenic determinants. Okay, the antigenic property of a particular antigen is determined by the presence of this epitope. Look at the option, you can see a term haptin. What are haptins? Haptins are low molecular weight molecule. They are small organic molecules that are antigenic but not immunogenic. They can act as antigen. Why? Because they contain the antigenic determinants, namely epitope, but they are totally incapable of provoking what? Immunological reaction. Okay, they can bind to immune cells but fail to induce humoral or cell mediated immune responses. Hence, no antibodies are raised against them. Correct answer for this question is option A, haptic. Next question is, cell splicing introns were first discovered in option A, Thermophilus aquaticus, option B, Trypanosoma brucei, option C, Trypanosoma gambians, and option D, Tetrahymena thermophila. You know that the process of production of RNA from DNA is known as transcription, okay, or RNA synthesis. But the RNA produced is a long RNA. It is known as heterogeneous nuclear RNA. It is the first product of transcription, primary product of transcription. Hence, that heterogeneous nuclear RNA is also known as what? Primary transcript. And that heterogeneous nuclear RNA or primary transcript contain both coding sequences as well as non-coding sequences. And that coding sequences are called as exons and non-coding sequences are called as introns. Okay, so the primary product of transcription contain two units, coding sequence namely exons and non-coding sequence namely introns. Hence that primary transcript is said to be heterogeneous. It's made up of two different units, isn't it? Coding as well as non-coding sequence. And that heterogeneous nuclear RNA is produced within the nucleus. Hence the name nuclear, heterogeneous nuclear RNA. Why that primary transcript is called heterogeneous nuclear RNA? Why it is called heterogeneous? Why? Because it is made up of two units, coding sequence namely exons and non-coding sequence namely introns. Why it is called nuclear RNA? Because that RNA is produced within the nucleus. Okay, that primary transcript is known as heterogeneous nuclear RNA, abbreviately known as hnRNA. You know that the process of production of protein from messenger RNA or mRNA is known as biosynthesis of protein or translation. To produce protein, this hnRNA should be converted into messenger RNA. In order to convert the hnRNA into messenger RNA, the non-coding sequences, namely intron present in hnRNA, should be removed. And the process of removal of these intron from the structure of hnRNA is known as splicing or RNA splicing. Okay, and this splicing is carried out by a specialized protein known as spliceosome. Generally, splicing occurs with the help of a group of protein or enzyme known as spliceosome. But in some organisms, the RNA itself can promote this splicing without the help of any protein or enzyme. Such a type of splicing is known as cell splicing. And this cell splicing introns were first discovered in a protozoan, namely Tetrahymena thermophila. So, answer for this question is option D, Tetrahymena thermophila. Remember, the cell splicing introns were first discovered in a protozoan, namely Tetrahymena thermophila. Here is your question. The question is, a protein kinase family involved in mitotic spindle formation. Option A, ATR. Option B, ATM. Option C, armadillo. Option D, aurora. Okay, let us understand the term protein kinase in brief. Okay, what do you mean by protein kinase? Protein kinase are enzymes capable of phosphorylating other proteins. As a result, that phosphorylated protein become activated or inactivated. What do you mean by phosphorylation? Addition of phosphate group to any compound can be called as phosphorylation. Usually, the phosphate group is provided by adenosine triphosphate or ATP. Okay, so the main function of protein kinase is to activate or inactivate other protein by phosphorylating. Okay, the major cellular process performed by protein kinase are signal transduction, the second one cell differentiation, third one cell proliferation, and also involved in cell cycle progression. Okay, enzyme having this activity, that is phosphorylating activity, thereby activating or inactivating a particular protein, comes under the category protein kinase family. Okay, the first option provided in the question is ATR. Okay, ATR is a serine threonine specific protein kinase enzyme. Okay, it's a protein kinase enzyme. Why it is known as serine threonine specific protein kinase? Because it is responsible for the phosphorylation of other protein, especially in the serine or threonine residue. Okay, this enzyme phosphorylated other protein on the OH group of amino acid residue, namely serine or threonine. Hence the name serine threonine specific protein kinase. Okay, the full form of this ATR is ataxia, telangiectasia and RAD3 related protein kinase. Okay, they are mainly involved in sensing DNA damage. Okay, and thereby activating the DNA damage checkpoint. That eventually leads to what? Cell cycle arrest. Okay, the main point is that ATR is activated in response to persistent single-stranded DNA, which is a common intermediate form during DNA damage detection and repair. Okay, so this protein kinase, ATR protein kinase, is mainly concerned with the sensing of what? DNA damage. Thereby it leads to what? Cell cycle arrest. Okay. The second option provided in the question is ATM. The full form of ATM is ataxia telangiectasia mutated protein kinase. Okay, and this protein kinase is activated whenever there is a DNA double strand break. Okay, and we can generally say that these ATM become activated when there is a genotoxic stress like double strand break of DNA. Okay, and these activation of these protein kinase, ATM protein kinase, is mainly for maintaining what? Cellular homeostasis. Okay, remember the ATR protein kinase is activated whenever there is a DNA damage, especially single strand break, whereas the ATM protein kinase is activated whenever there is a genotoxic stress, namely double strand break, DNA double strand break. Okay. Okay, the third option in the question is armadillo. Okay, armadillo is a protein produced by a gene, armadillo gene. It is a segment polarity gene found in Drosophila, mainly involved in transduction. Okay, actually this armadillo protein produced by armadillo gene mainly determines the polarity of segments in Drosophila. As far as your CSI net exam is concerned, this Drosophila developmental biology, especially the protein involved in the anterior posterior axis development, as well as the segment determining protein, is of much significance. Okay. The last option in the question is Aurora. Aurora is a serine threonine specific protein kinase. They are mainly involved in cell proliferation. Okay, they transfer the genetic materials to a daughter cell, and the most specific function of the aurora is what? Spindle formation. Okay, they are involved in the spindle fiber formation during mitosis, which enable the transfer of chromatids to the daughter cell, which eventually leads to what? Cell division. Okay, I think you people got some idea about all these options mentioned here. Now you know the correct answer for this question, and the answer for this question is option D, aurora. Remember, aurora is mainly involved in mitotic spindle formation, which enables the proper cell division. The next question is, elaioplast is a plastid that stores option A lipid, option B protein, option C carbohydrate, and option D chlorophyll. Okay, let us discuss some important points regarding plastids. Plastids are double membrane bound cell organelle commonly found in plants which either store pigment or nutrients. Okay, plastids can be classified into three types, and they are chloroplast, chromoplast, and leucoplast. Chloroplasts are green colored plastids, and the green color is due to a pigment known as chlorophyll. Chromoplasts are colored plastid other than green color. The common pigment present in chromoplasts are carotene and xanthophyll. Carotenes are yellow to orange color pigment, whereas xanthophyll are yellow color pigment. Okay, Leucoplasts neither stores any pigment. Instead of pigments, they stores nutrients. So, nutrient storing plastids can be called as leucoplasts. Leucoplasts are further divided into three types, and they are amyloplast, alluroplast, and eleoplast. Amyloplast is the type of leucoplast which stores carbohydrates such as starch, whereas alluroplast is a type of leucoplast which stores protein. And eleoplast is also a type of leucoplast which stores oil or fat. The example for amyloplast is potato. The example for alluroplast is the maize. Okay, when you are analyzing maize, you can see a layer known as alluron layer, that protein-rich layer containing a leucoplast, namely alluroplast. Okay, eleoplast is commonly found in tuberose and castor. Okay. Remember, amyloplast stores starch, alluroplast stores protein, eleoplast stores oil or fat. Okay. So the correct answer for this question is option A, lipid. You know that eleoplasts are type of leucoplast which stores either fat or oil. Both fat and oil comes under the category lipid. Hence the answer is option A, lipid. The next question is attenuation involves A. Enhancing the virulence and antigenic property, B. Diminishing the virulence and retaining the antigenic property, C. Diminishing the virulence and antigenic property, D. Enhancing the virulence and diminishing the antigenic property. Okay, let us understand the term attenuation in brief. Attenuation is a term which is related to immunology. Attenuation is a process in which a pathogen or antigen is strong enough to cause immune response but too weak to cause disease. Okay. As a result of attenuation process, a particular pathogen or antigen is having the capacity to cause immune response, but it is too weak to generate a particular disease. Okay. Attenuation stimulates an excellent immune response that is nearly as good as compared with the infection of wild type pathogen, but as a result of attenuation process, that particular pathogen is totally incapable of generating a particular disease. Actually, attenuation diminishes the virulence or disease causing capacity of pathogen but retains its antigenic property, that is, retains its property to provoke immunological response. Okay, and this attenuation process acts as one of the basic principles by which vaccines are developed. Such a type of vaccines are said to be attenuated vaccines. Okay, you know that the father of vaccination is considered as Edward Jenner, isn't it? But the basic principle of vaccination was first developed by Louis Pasteur. Remember, okay, Edward Jenner is considered as the father of vaccination, but the basic principle of vaccination was first developed by, not by Edward Jenner, it is by whom? It is by Louis Pasteur. Okay. I think now you got the correct answer. Yes, the correct answer for this question is option B, diminishing the virulence and retaining the antigenic property. The next question is, for renal medulla, brain, etc., sole source of metabolic energy is option A, Krebs cycle, option B, ornithine cycle, option C, EMP pathway, and the last option is electron transport series. The breakdown of cis-carbon molecule glucose into two molecules of three-carbon molecules, namely pyruvic acid, is known as glycolysis or glycolytic breakdown. This glycolysis is also known as EMP pathway because various steps in this pathway were discovered by three scientists, namely Emden, Meyerhof, and Parnas. In order to honor these scientists, this pathway was provided with the first letters of these three scientists, namely EMP pathway. Okay, and this glycolysis process is acting as the sole source of metabolic energy in some mammalian tissue and cell type. For example, erythrocyte, renal medulla, brain, sperm, etc. Okay, some plant tissue that are modified to store starch, such as potato tubers, and some other aquatic plants derive most of their energy from glycolysis. Many anaerobic microorganisms are entirely dependent on glycolysis. So the correct answer for this question is option C, EMP pathway. EMP pathway is also known as glycolysis or glycolytic breakdown. Okay. Okay, let us look at the other option also. Okay, the first option, Krebs cycle. Okay, what do you mean by Krebs cycle? Whatever the pyruvic acid formed in glycolysis enters the Krebs cycle. Okay, in Krebs cycle, each molecule of pyruvic acid get converted into carbon dioxide plus water plus energy. And Krebs cycle occurs in mitochondrial matrix, whereas glycolysis or EMP pathway occurs in cytoplasm or cytosol. Okay, what do you mean by ornithine cycle? Urea cycle is also known as ornithine cycle. In ornithine cycle, the toxic ammonia is get converted into less toxic urea. Okay, this ornithine cycle or urea cycle is exhibited by ureotelic animals. Okay, and ornithine cycle usually occurs in the mitochondria of liver cell. Then what do you mean by electron transport series? This electron transport series is also known as electron transport chain, ETC, or electron transport system, like that. Okay, in electron transport system, the oxidative phosphorylation or terminal oxidation takes place. It is the last or final step of aerobic respiration. What is the main function of electron transport series? In electron transport series, the high energy compounds such as NADH and FADH2 are converted into their respective ATP. Okay, when NADH get enter into electron transport series, one molecule of NADH will produce 3 molecules of ATP and one molecule of FADH2 will produce 2 molecules of ATP. Okay, you know that NADH and FADH2 are the reduced form of NAD and FAD respectively. What is the full form of NAD? Nicotinamide adenine dinucleotide. What is the full form of FAD? Flavin adenine dinucleotide. Okay, these are also coenzymes. Okay, and these coenzymes, namely NAD and FAD, are derived from vitamin B complex. Okay, NAD is derived from a vitamin, what? Niacin, whereas the FAD is derived from a vitamin riboflavin, namely vitamin B2. Okay, so the correct option for this question is option C, EMP pathway. EMP pathway is also known as glycolysis. Why glycolysis is known as EMP pathway? Because various steps in this cycle were discovered by 3 scientists, namely Gustav Emden, Otto Meyerhof and J. Parnas. Emden, Meyerhof and Parnas. Okay. In order to honor their name, the first letter of their name is indicated by the pathway, namely EMP pathway. The next question is, which of the following is excretory in function? Option A, proamnion. Option B, chorion. Option C, allantois. Option D, amnion. In the question, the first option is proamnion. Okay. In the embryo stage, around 20 hours after fertilization, the head fold is visible as a half-circular elevated structure in the front of germ layer. The proamnion lies in front of this head fold as a clear zone. The amnion is one of the membranes that will protect the embryo in a later stage. So, from the proamnion, this amnion is developing. Okay. So, the main function of the proamnion is to derive as amnion, thereby providing protection. Okay. B option in the question is chorion. The chorion and the amnion together form amniotic sac. It is formed by extra-embryonic mesoderm and the 2 layers of trophoblast that surround the embryo and other membranes. The chronic villi emerge from the chorion, invert the endometrium and allow the transfer of nutrients from the maternal blood to fetal blood. Okay, so the chronic villi emerge from the chorion, okay, thereby providing nutrients. So the main function of the chorion is to provide or providing a facility for nutrients. Okay. The main function of allantois is to collect liquid waste from the embryo as well as to exchange gas used by the embryo. Okay, hence allantois is excretory in function. Okay, let us conclude all the options once again. Okay, proamnion is the region from which the amnion is developed or derived. Okay, and the main function of amnion is to provide protection, and the main function of chorion is to provide nutrients through a chronic villi. Then the main function of allantois is to perform excretion. It is excretory in function. Okay. So the correct answer for this question is option C, allantois, which is excretory in function. Question is, dicamarol function as an anticoagulant by option A, bringing about vitamin K deficiency, option B, removing calcium ions, option C, destroying fibrinogen, and the final option is facilitating the action of antithrombin. Let us understand the term dicamarol in brief. Dicamarol is also known as dicumarol. It is a hydroxycoumarin. It is a naturally occurring oral anticoagulant drug that depletes the store of vitamin K. Okay, you know that vitamin K is one of the important vitamin essential for the normal blood clotting process. Okay, so this dicamarol acts as anticoagulant by interfere with what vitamin K metabolism process. Okay, why this vitamin K is important for blood clotting? Why? Because this vitamin K synthesizes a lot of clotting factor for the normal blood clotting process. Okay, when there is a deficiency of vitamin K, it will adversely affect the normal process of blood clotting. Okay, you know that any substance which has the capacity to inhibit normal blood clotting can be called as anticoagulant. Okay, so my point is that dicamarol acts as anticoagulant by depleting vitamin K synthesis, isn't it? Okay. And this dicamarol, I already said that it is a hydroxycoumarin originally isolated from molding seed clover hay plant. Okay, let us go through the options once again. Yes, I think you people got the correct answer just now. Yes, the answer is what? The option A, bringing about vitamin K deficiency. Dicamarol function as an anticoagulant by bringing about vitamin K deficiency. So the correct answer for this question is option A. The next question is reverse transcriptase is option A, DNA dependent RNA polymerase, option B, DNA dependent DNA polymerase, option C, RNA dependent RNA polymerase, and option D, RNA dependent DNA polymerase. Okay, what do you mean by RNA polymerase? RNA synthesizing enzyme is known as RNA polymerase. The process of production of RNA from DNA is known as transcription. Okay, so RNA synthesis can be also called as transcription. Hence, RNA polymerase is acting as a key enzyme of transcription. You know that RNA is synthesized from DNA by the enzyme RNA polymerase. Hence, RNA is synthesized in accordance with the sequences found on the DNA. So, RNA polymerase synthesizes RNA by looking at the sequences found in DNA. Hence, that RNA polymerase is known as DNA dependent RNA polymerase. Okay. Hence, we can say that the key enzyme of transcription is DNA dependent RNA polymerase. The key enzyme of transcription, namely DNA dependent RNA polymerase, can be also called as transcriptase. Okay, so you can expect a question like that. Transcriptase is, the answer is transcriptase is DNA dependent RNA polymerase. In transcription, RNA is synthesized from DNA. Then what do you mean by reverse transcription? The process of synthesis of DNA from RNA is known as reverse transcription. It is just a reverse to transcription. Okay, So in reverse transcription, what happens? DNA is synthesized from RNA. Okay, DNA is synthesized by looking at RNA, isn't it? Okay. Then DNA synthesizing enzyme are known as DNA polymerase. In reverse transcription, DNA is synthesized by DNA polymerase by looking at what? RNA. Hence, the key enzyme of reverse transcription is what? RNA dependent DNA polymerase. It is also known as what? Reverse transcriptose. Then what do you mean by DNA replication? The process of production of daughter DNA from parent DNA can be called as DNA replication or DNA duplication or DNA synthesis. Okay, in DNA replication, DNA is synthesized from parent DNA. So, the enzyme is what? DNA polymerase. In DNA replication, DNA polymerase synthesizes daughter DNA by looking at the sequences of the parent DNA. So, we can called as the key enzyme of DNA replication is DNA dependent DNA polymerase. Once again, I am repeating, remember the key enzyme of transcription is DNA dependent RNA polymerase. The key enzyme of reverse transcription is what? RNA dependent DNA polymerase. The key enzyme of DNA replication is DNA dependent DNA polymerase. Remember, the reverse transcription is also known as what? Terminism. Okay, reverse transcription is commonly shown by viruses having RNA as genome. Okay, so the correct answer for this question is what? The reverse transcriptase is RNA dependent DNA polymerase. So the correct answer for this question is option D. Okay, I think we missed the question number 32. So I am discussing the question number 32 after the question number 33. The question is, the disease due to nitrogen toxicity is option A, Kerson's disease, option B, Addison's disease, option C, Alzheimer's disease, and the last option is Wilson's disease. Okay, the first option provided in the question is Kerson's disease. Let us understand some important point regarding Kerson's disease in brief. The Kerson's sickness is also known as decompression sickness and is also known by another name, diver's disease. Why? Because it is commonly exhibited by those persons performing deep diving. Okay, and this Kerson's disease occurs when the body is exposed to sudden drop in surrounding pressure. Okay, most frequently during deep sea diving or when flying in a non-pressurized aircraft. This Kerson's disease is mainly caused due to the formation of gas bubbles in the blood and tissue. At normal altitude, nitrogen and other gases are exhaled or dissolved in the blood and tissue. However, during severe changes in altitude and air pressure, nitrogen and other gases form gas bubbles. These bubbles block the flow of blood, and this condition may lead to nitrogen toxicity and it may be fatal. Okay, and it leads to death if it is not properly treated. So the important point is that the Kerson's disease is mainly due to nitrogen toxicity and commonly exhibited by divers. The B option in the question is Addison's disease. Addison's disease is also called as adrenal insufficiency. It is not a common disorder. That occurs when body doesn't produce enough of a certain hormone. Okay, in Addison's disease, the adrenal gland produces too little cortisol and often too little aldosterone. The C option in the question is Alzheimer's disease. It's a progressively degenerative neurological disorder leading to dementia, memory-related problems, with increasing mental confusion, emotional instability, and premature death. It's a A common disease. Okay, very familiar disease. Okay, the option D in the question is Wilson's disease. Wilson's disease is an autosomal recessive disorder. It is characterized by low ceruloplasmin. Ceruloplasmin is nothing but it is a protein, a major copper carrying protein found in the blood. Okay, this disease is also characterized by higher copper deposition in liver, brain, kidneys, eyes, heart, and also it is characterized by a condition called hemolysis. So the correct answer for this question is option A, Caisson's disease. Caisson's disease is due to what? Due to nitrogen toxicity. Let's move to the next question. The question is a sex-linked recessive disorder in which the affected children mutilate themselves. Okay, mutilate means harm themselves. Okay, option A, Cray-Duchat syndrome. Option B, Patau syndrome. Option C, Lesch-Nyhan syndrome. And the last option is Tay-Sachs syndrome. Let us move to the first option, Cray-Duchat syndrome. The word Cray-Duchat means the cat's cry. The Cray-Duchat syndrome is also known as chromosome 5p deletion syndrome. You know that this Cray-Duchat syndrome is due to a deletion of a chromosomal fragment in the short arm of the fifth chromosome of human. Okay, here P means what? You know that in chromosome there are certain long arm and short arm. The long arm of chromosome is represented by the letter, small letter Q, and the short arm of the chromosome is represented by the small letter P. Okay, the Cray-Duchat syndrome is due to a chromosomal aberration, namely deletion in the short arm of fifth chromosome pair. Okay, you know that chromosomal aberrations are a type of chromosomal mutation due to change in the structure of chromosome. Okay, this Cray-Duchat syndrome is a typical example for chromosomal aberration, namely deletion. The term Cray-Duchat is a French term which means cat cry or call of the cat, referring to the characteristic cat-like cry of affected children. Those children having this disease produce meow-like sound, cat cry-like sound. Okay. Then why those children produce characteristic cat-like cry? It is due to a problem within the larynx. That is, the larynx is not properly developed in those affected children. Okay, the B option in the question is Patau syndrome. It is due to the trisomy of chromosome 13. Okay, so this Patau syndrome is also known by the name what trisomy D or trisomy 13. What do you mean by trisomy? When an extra chromosome is added to a chromosome remain in a diploid condition. That is known as trisomy. 2n plus 1 is known as trisomy. Okay, and this trisomy 13 is caused by non-disjunction of chromosome during meiosis. What do you mean by non-disjunction? Failure of separation of chromosome during cell division can be called as non-disjunction. Okay, but it affects about 1 in 12,000 live births. Okay, it's a rather rare disease. More than 80% of infants with Patau syndrome die within their first year of life. The C option in the question is Lesch-Nyhan syndrome. It is also known as Nyhan syndrome, Kelly-Sigmiller syndrome, and also by the name juvenile gout. It's a hereditary disorder of purine metabolism, characterized by mental retardation and also characterized by self-mutilation or self-harming of the fingers and lips by biting, impaired renal function, and abnormal physical development. Okay, of which self-mutilation is a easily observed symptom of this disorder. Okay, it's a recessive disease that is linked to X chromosome. Okay, it is due to the deficiency of an enzyme known as hypoxanthine guanine phosphoribosyl transferase. It is an essential enzyme involved in purine metabolism. The major symptoms of this disease include overproduction of uric acid, urate crystal formation which look like orange sand, which are deposited in diapers of the babies, then kidney stone, blood in the urine, then the condition named dysphagia, difficulty in swallowing, then swelling of the joint, then vomiting, like that. And the behavioral abnormality exhibited by those individuals having this disease include impaired cognitive function, ill-developed cognitive function. Then the mostly visible symptom namely self-mutilation, harming, self-harming. Self-mutilation means self-harming. Then aggression, impulsion, like that. The D option in the question is Tay-Sachs disease or Tay-Sachs syndrome. Tay-Sachs syndrome is a rare disorder passed from parents to child. It is due to the absence of an enzyme that helps break down the fatty substance called the gangliosides. You know that ganglioside is a compound lipid. It is a glycolipid. You know that. Okay. The build-up to toxic level in the child's brain and affect the function of the nerve cell. This ganglioside accumulate in the child's brain at toxic level and affect the function of the nerve cell. This Tay-Sachs syndrome is a fatal or a serious genetic disorder most commonly occurring in children. Results in progressive destruction of the nerve system due to the accumulation of compound lipid or a glycolipid namely gangliosides. Okay, it is due to the absence of a vital enzyme called hexosaminidase. This hexosaminidase or hex A is essential for this ganglioside metabolism. Okay. Okay, I hope now you people got the correct answer for this question. Yes, the correct answer for this question is option C, Lesch-Nyhan syndrome. Lesch-Nyhan syndrome is characterized by self-mutilation. Let's move to the next question. Which of the following is an ideal substance for measuring GFR? Option A, glucose. Option B, inulin. Option C, creatinine. And option D, albumin. When you are analyzing this question, you can see a term GFR. The full form of GFR is glomerular filtration rate. Glomerular filtration rate is a test used to check how well your kidneys are working. Okay, it estimates how much blood passes through glomeruli per minute. You know that glomeruli are tiny filters in the kidneys that filter waste from the blood. Okay, and the ideal substance which is used for measuring GFR is a polysaccharide, namely inulin. Okay, what do you mean by polysaccharide? Polysaccharides are carbohydrate made up of several monosaccharide units. Okay, what do you mean by monosaccharide? Monosaccharides are simplest carbohydrate which cannot be further hydrolyzed into simpler units. Okay, so the polysaccharide inulin is made up of monosaccharide, namely fructose. In inulin, fructose are held together by a glycosidic bond known as beta-2,1 glycosidic bond. You know that glycosidic bonds are the characteristic bond found in carbohydrate. Okay, this insulin can be also seen the root tuber of dahlia. Okay, as far as your examination is concerned, the bonds found in carbohydrates are of much importance. So you have to study all the bond found in carbohydrate. Okay. Okay, I will try to upload a video regarding the various bonds found in carbohydrate because it is very important as far as your examination is concerned. Okay. So the correct answer for this question is option B, inulin. Inulin is a polysaccharide made up of monosaccharide units, namely fructose. Aerobic breakdown of 6-carbon glucose yields 38 ATP. How many ATP molecules are produced by a fatty acid having 18 carbon? And the options are A 114, B 147, C 48 and D 76. This question is about the breaking down of fatty acid. Okay, and they asked about how many ATP molecules are produced by a fatty acid having 18 carbon. Okay, there is no relevance for the first part of the question. That is, aerobic breakdown of 6-carbon glucose yield 38 ATP. And they provided that part in the question not to confuse you people. Okay, so we have to concentrate on the second part of the question, that is how many ATP molecules are produced by a fatty acid having 18 carbon. Okay, you know that the process of breakdown of fatty acid is known as beta oxidation of fatty acid. Okay, and this beta oxidation of fatty acid mainly takes place in mitochondrial matrix. Okay, but the activation of fatty acid takes place in the cytoplasm or cytosol. Consider if a fatty acid having 8 carbon atom undergo beta oxidation. In the first round of beta oxidation, what we will get? We will get a 2 carbon acetyl-CoA and 6 carbon fatty acyl-CoA. Okay, then what happens? This 2 carbon acetyl-CoA enters the Krebs cycle or citric acid cycle or TCA cycle and produces 12 ATP. You know that when an acetyl-CoA enters the Krebs cycle, a single round of Krebs cycle will produce how many ATP? Remember, 12 ATP molecule. Okay, once again I am telling, when a fatty acid having 8 carbon atom undergo beta oxidation of fatty acid. In the first round of cycle, what happens? This fatty acid having 8 carbon atom breaks into 2 carbon containing acetyl-CoA and 6 carbon containing fatty acyl-CoA, isn't it? And this 2 carbon containing acetyl-CoA enters the Krebs cycle and produce 12 ATP molecule, and the remaining 6 carbon containing fatty acyl-CoA again undergo the next round of what? Beta oxidation of fatty acid. Okay, when that 6 carbon containing fatty acyl-CoA undergo beta oxidation of fatty acid, will yield what? Will yield 2 carbon acetyl-CoA and 4 carbon fatty acyl-CoA. Okay, this 2 carbon acetyl-CoA again enter the Krebs cycle and will yield 12 ATP molecule. Okay, and the 4 carbon fatty acyl-CoA again undergo beta oxidation of fatty acid and will yield 2 carbon acetyl-CoA plus again 2 carbon acetyl-CoA. Okay, and these two molecules of acetyl-CoA again undergo Krebs cycle. Each molecule will produce 12 ATP. Okay. My point is that fatty acid will undergo beta oxidation of fatty acid until it breaks into 2 molecules of 2 carbon acetyl-CoA. Okay, the fatty acetyl-CoA get converted finally into what? 2 carbon acetyl-CoA, and this acetyl-CoA again enter the Krebs cycle. Okay. Okay, I am asking you a question, that is, when a 8 carbon containing fatty acid undergo beta oxidation of fatty acid, how many cycle of beta oxidation will occur? How many? At first the 8 carbon containing fatty acid undergo beta oxidation to produce 2 carbon acetyl-CoA and 4 carbon fatty acyl-CoA. Okay, that is one cycle. Next one, this 6 carbon fatty acyl-CoA again undergo the second round of beta oxidation of fatty acid to produce 2 carbon acetyl-CoA and what? 4 carbon fatty acyl-CoA. Again, this 4 carbon fatty acyl-CoA undergo the third round of beta oxidation of fatty acid to give what? 2 molecules of acetyl-CoA, that is acetyl-CoA plus acetyl-CoA. So, when a 8 carbon containing fatty acid undergo beta oxidation of fatty acid, it will perform how many round of beta oxidation fatty acid cycle? 3 round of beta oxidation fatty acid cycle. So, likewise we have to calculate, that is in the question they are asking about an 18 carbon containing fatty acid undergo breakdown, that is beta oxidation of fatty acid. Then how many cycle it will perform, how many cycle of beta oxidation fatty acid will perform by that 18 carbon containing fatty acid? Calculate. Suppose I am giving you a chain containing 18 beads. you have to break that chain into fragments containing 2 bits only. Then in order to convert that long chain into into fragments containing 2 bits, how many times you people have to break the chain? You got it? Yes. 8 times you have to break down that chain to get fragments of 2 bits. Okay. Likewise, in order to break that 18 carbon containing fatty acid, how many round of beta oxidation of fatty acid will occur? Absolutely. 8 round of beta oxidation of fatty acid will occur. Remember. Single round or a single cycle of beta oxidation of fatty acid will produce one FADH2 and one NADH. You know that one FADH2 on electron transport chain will produce what? 2 molecules of ATP. Okay, and one NADH will yield how many ATP? 3 molecules of ATP. Remember, one FADH2 undergo electron transport chain to produce 2 ATP and one NADH will produce 3 ATP. You know that these FADH2 and NADH are high energy compounds. That's why they are producing more than one ATP. Okay, but according to modern energy concept, one FADH2 will produce 1.5 ATP and one NADH will produce 2.5 ATP. But all the concept, one FADH2 will produce 2 ATP and one NADH will produce 3 ATP. Remember this point. Okay. Okay, in beta oxidation fatty acid, at first the fatty acid should be activated. And this activation of fatty acid takes place in the cytoplasm or cytosol. Remember, not in the mitochondria. Okay, and the activation of fatty acid requires 2 ATP. That is, 2 ATP molecules are consumed for or utilized for the activation of fatty acid. And this activation of fatty acid takes place in where? Cytoplasm or cytosol. These activated fatty acids are then transported from the cytoplasm to the mitochondrial matrix by a transport system known as carnitine transport system. So remember, for the activation of fatty acid, 2 ATP molecules are consumed. Okay. So when we are going to calculate the ATP molecules from the fatty acid, we have to consider this also. Here 2 ATP molecules are consumed. How these 2 ATP molecules are consumed? Okay, you know that in the activation step, the fatty acid in the cytoplasm react with the 1 molecule of ATP. Okay, but in the 1 molecule of ATP, from the 1 molecule of ATP, 2 molecules of phosphates are added to where? The fatty acid. Okay, as a result, that 1 molecule of ATP is converted into AMP, adenosine monophosphate. Okay, adenosine monophosphate then again convert into what? ADP. For that process, 1 ATP is also utilized. So, whenever ATP get converted into AMP, it is equivalent to the consumption of 2 ATP molecules. Okay. Okay. That's why in the activation step, that is fatty acid activation step, 2 ATP molecules are consumed or utilized. Okay, I think now you got the basic idea to calculate ATP. Okay. Okay, so ATP produced by the breakdown of fatty acid having 18 carbon atom. Okay, you know that the fatty acid having 18 carbon atom is nothing but it is stearic acid. Stearic acid. Okay, how many round of beta oxidation cycle will that stearic acid will perform? 8 round. Okay, so number of beta oxidation cycle is equal to 8. Okay, 1 round of beta oxidation cycle will produce, you know that, 1 FADH2 and 1 NADH, isn't it? So 8 round of beta oxidation cycle will produce 8 FADH2 and 8 NADH. Okay, 8 FADH2 is equal to 16 ATP, 8 NADH is equal to 24 ATP. Okay, because 1 FADH2 is equal to 2 ATP, 1 NADH is equal to 3 ATP. Okay, then fatty acid with 18 carbon atom will produce how many acetyl-CoA at last? It will produce 9 acetyl-CoA, and each and every acetyl-CoA will enter into Krebs cycle. So, as soon as this 9 acetyl-CoA is formed, this 9 acetyl-CoA enter the Krebs cycle, and each round of Krebs cycle will produce how many ATP? 12 ATP. So, 9 acetyl-CoA will produce 9 into 12, 108 ATP. So, the total ATP produced by the breakdown of 18 carbon fatty acid is 148 ATP. Okay, but from this 148 ATP, you have to reduce 2 ATP. Why? Because the 2 ATP is consumed for the activation of fatty acid. Remember, okay, so 148 minus 2, the answer is 146 ATP. Okay. Okay, look at the question at option once again. Okay, we have got the answer 146, but in the option you can find only 147. Why? Because in the older concept, for the activation of fatty acid, only 1 ATP is required. Okay, that is the older concept. Okay, but nowadays we know that for the activation of fatty acid, 2 ATP molecules are consumed or utilized. So you have to reduce or subtract 2 ATP molecule from the gross ATP, that is 148. Okay, so as per the new concept, the correct answer is 146. Anyway, the answer given by PSC is B option, that is 147. Okay, I think you people now familiar to calculate such a type of question, that is how many ATPs are produced from the fatty acid. You have to go through the entire video, then you will get somewhat idea about to calculate the ATP from fatty acid having any carbon number. Okay, soon I will upload the carbon number of fatty acid, easy way to study the carbon number of fatty acid, and the entire cyclo-beta oxidation after the discussion of the previous year question. Okay. So, for this question, the most appropriate answer is the option B. Okay. Anyway, the accurate answer is not 147, is it? It is 146. Okay. Hi, my dear friends. Welcome to my channel, Biology for All. Today I am going to discuss the 9th part of previous year question for the post scientific biology, Forensic Science Laboratory. Let's start. Okay, here is your question. Which of the molecular event is not occurring as a result of capacitation of sperm? Option A, increased sperm metabolism and motility. Option B, increase in cyclic AMP production. Option C, increase in membrane potential of sperm. Option D, decrease in cholesterol level. Okay, what I mean by sperm capacitation or capacitation of sperm? Okay, in order to penetrate and fertilize an egg, the sperm should undergo certain physiological and biochemical changes. Okay, and these physiological and biochemical changes undergone by spermatozoa in order to have the ability to penetrate and fertilize an egg is called as sperm capacitation or capacitation of sperm. Okay. Or simply we can define the sperm capacitation as functional maturation of spermatozoa. Okay, when the spermatozoa get functionally maturated, then only it is able to penetrate and fertilize an egg. Okay, that is called as sperm capacitation. Okay, let us understand the major molecular events occur during the capacitation of sperm. First one is a destabilization or structural disintegration of acrosomal sperm head membrane, okay, which help the sperm to penetrate the outer layer of the egg. Next one is a chemical change that occur in the tail region of the sperm. As a result, the sperm become more and more activated. Okay, at that time we can see the more movement of hyper movement of the sperm. Okay, then how it happens? Okay, these changes are facilitated by the removal of certain sterols. Okay, You know that cholesterol is a derived lipid coming under the category sterol. Whenever these sterol, namely cholesterol and certain glycoprotein are removed from the membrane, as a result, increased membrane permeability can be seen. Remember, increased membrane permeability means what? What? Reduction in membrane potential, not increase in membrane potential. Okay, whenever there is increased permeability, you can see that reduction in membrane potential. That is, the membrane potential is negative, not positive, remember. Okay, as a result of when this membrane become more and more permeable, as a result, more amount of calcium influx can be seen. More amount of calcium enter into the sperm cell. Okay. As a result of increased influx of calcium 2 plus ion, which will activated what? The secondary messenger, namely cyclic AMP. You know that cyclic AMP is a secondary messenger. Okay. Which will ultimately result the hyperactivation sperm. So in general, we can say that during capacitation of sperm, increased sperm metabolism and motility can be seen. Increase in cyclic AMP production can be seen. And decrease in the cholesterol level. Steroids such as cholesterol become reduced in order to increase the increase the permeability of the membrane. Whenever the permeability of the membrane become increase, decrease in membrane potential of sperm can be seen. Okay, not increase in membrane potential of sperm. Right. Okay, now I think you people got the correct answer just now, isn't it? The correct answer is what? Option C, increase in membrane potential of sperm. Okay, the membrane potential of sperm become reduced during capacitation of sperm. That's why that membrane of sperm is exhibiting more permeability. Okay, so the event which is not occur during the capacitation of sperm is the option C, increase in membrane potential of sperm. What is the correct event taking place during that time? That is decrease in membrane potential of sperm. So the correct answer for this question is option C, increase in membrane potential of sperm. Okay. When your CSIR net examination is concerned, the prevention of polyspermy in sea urchin is very important. Okay, so you have to study that portion also if you are preparing for the CSIR UGC net license. Okay. Okay, next question is the basic cause of senile dementia as well as Alzheimer's disease is option A, accumulation of acetylcholine mimics, B, accumulation of adrenaline mimics, C, degeneration of cholinergic nerve terminals, and the last option is degeneration of adrenergic nerve terminals. Okay, you know that both Alzheimer's disease and dementia are characterized by severe memory loss. Okay, let us understand the actual reasons of these two diseases. Okay, that is, two neurotransmitters play an important role in Alzheimer's as well as dementia. And the neurotransmitters are acetylcholine and glutamate. You know that glutamate or glutamic acid is an acidic amino acid. Okay. Acetylcholine activates muscles and helps with arousal, short-term memory and learning. Okay. These acetylcholine activate certain muscles related to arousal, then it enhances short-term memory, then learning ability, like that. Okay, individuals with Alzheimer's disease have low level of acetylcholine. Very important point. Okay, those individuals having this Alzheimer's disease as well as dementia are provided with very low level of acetylcholine. Okay. Some research suggests that the plaques may be one of the reasons for low level of acetylcholine because they increase the activity of chemical called acetylcholine esterase, which is involved in the breaking down of acetylcholine. Okay. Some recent research suggests that certain plaques which is formed will activate the acetylcholine esterase enzyme. This acetylcholine esterase enzyme is mainly concerned with the breaking down of acetylcholine. Thereby, it will cause considerable reduction in the acetylcholine concentration, isn't it? Too much acetylcholine esterase has the overall effect of decreasing acetylcholine level, isn't it? Which contributes to the characteristic symptoms of Alzheimer's disease as well as dementia. Okay, when the cell contain more amount of acetylcholinesterase enzyme, it will degrade more amount of acetylcholine that will result the disease, namely Alzheimer's disease as well as dementia. Okay, and the degeneration of cholinergic nerves terminals or nerve endings also causes decrease in the level of acetylcholine. Okay, you know that these cholinergic nerve terminals are mainly concerned with the production of what? Acetylcholine. They passes nerve impulse through the agency of acetylcholine. When there is destruction or considerable damage to these cholinergic nerve terminals, a reduction, considerable reduction in the acetylcholine can be seen, which will ultimately leads to a disease called Alzheimer's disease or a disease called dementia. Okay. Okay, let us look at the options again. Now you people got the answer. The correct answer for this question is what? The option C, degeneration of cholinergic nerve terminals. Okay. The next question is, which of the following is not an amino acid derivative? Option A, adrenaline. Option B, calcitonin. Option C, norepinephrine. Option D, thyroxine. If a hormone is an amino acid derivative or derived from an amino acid, its chemical name will end in INE. It is a trick to find out amino acid derived hormone. Okay, so you have to remember this statement only. Those hormone derived from amino acid will have a suffix INE, not simply IN. Okay. Okay, look at the option. The hormone adrenaline, another name epinephrine, isn't it? Then norepinephrine, another name noradrenaline, and thyroxine. All these hormones are derived from amino acid. Some modification may take place in this in the particular amino acid, and by that modification these hormones are produced, isn't it? Normally adrenaline or epinephrine, norepinephrine or noradrenaline, and thyroxines are produced from an aromatic amino acid, namely tyrosine, Tyrosine, tyrosine. Okay, tyrosine is an aromatic amino acid with hydroxide or OH group. Okay, but when we are analyzing the calcitonin, we can see that calcitonin is a peptide hormone made up of 32 amino acid. In calcitonin, amino acid never undergo any modification. Amino acid get arranged together to form this calcitonin hormone. So remember, even though the calcitonins are made up of 32 amino acid, but they are not derived from amino acid. These 32 amino acids held together to form a polypeptide which is acting as a hormone called as calcitonin. But in the case of adrenaline, norepinephrine and thyroxine, they are derived from amino acid, namely tyrosine. Okay, I think now you got the correct answer. That is the hormone which is not an amino acid derivative is the option B, calcitonin. Okay, I also discussed a trick to find out this one, that is the amino acid derived hormone will have a suffix what? INE. Okay, in adrenaline, thyroxine and norepinephrine you can see INE, but in the case of calcitonin you can see only IN. Okay, so the correct answer is option B. The next question is the law which states that a condition which is found to be X chromosome linked in one mammal can be expected to be X chromosome linked in other mammals also is A. Owen's law, B. Muller's law, C. Calvin's law, and the last option is Bridges' law. Let us understand Owen's law in brief. Owen's law was proposed by a Japanese American biologist, Susumu Ono. This law states that the gene content of the mammalian species has been conserved over species not only in the DNA content but also in the genes themselves. Okay, According to Ono, the DNA content as well as the gene content conserved from species to species. Okay, that is, nearly all mammalian species have conserved the X chromosome from their primordial X chromosome of a common ancestor. Okay, that is, the X chromosome as well as genes present in the X chromosome found in all mammal is came from a common ancestor. Okay, that is, that particular X chromosome and its associated genes are highly conserved. Okay, and that law is known as Ono's law. So the correct answer for this question is option A, Ono's law. Okay, the remaining three laws are not related to biology, so I am not discussing those laws. Okay, inheritance of which of the following shows crisscross pattern? Option A, Lesch-Nyhan syndrome. Option B, color blindness. Option C, hemophilia. And option D, all the above. Let us understand crisscross inheritance in brief. Okay, what do you mean by inheritance? The phenomenon of transfer of characters from parents to offspring or young one can be called as inheritance, and such characters are called as inherited character. Okay, what do you mean by crisscross inheritance? When an inheritance is said to be crisscross inheritance, it should satisfy two criteria. The first criteria, that character should be transferred from a mother to son or from father to daughter. Okay, the second criteria is that character transferring mother or father should be a carrier. What do you mean by a carrier? Carriers are those individuals having a particular gene capable of causing a particular disease, but that gene is not expressing in them. Why? Because that character is in which condition? In heterozygous condition in them. Okay, so that they are called as carrier. Okay, remember, carriers are those individuals having a disease causing gene, but that disease causing gene is not expressing in them. Why? In order to express that particular character, the character coding gene should be in homozygous condition. But in that carrier persons, that disease causing gene is in heterozygous condition. That's why they are not expressing that particular disease. Okay. So, as far as the crisscross inheritance is concerned, when a character is transferred from mother to daughter or from father to son, such a type of character cannot be called as crisscross inheritance. If you want to call that particular character as crisscross inheritance, it should be from a father to daughter or from a mother to son. Okay, hemophilia, color blindness, Lesch-Nyhan syndrome, all these diseases are good example for crisscross inheritance. Okay, you know that all these genes are X-linked, all these diseases are X-linked genetic disorders. What do you mean by X-linked genetic disorder? Okay, you know that in our chromosome, we can see the gene which control body character and also the gene which control our sexual character. Those genes which control our body character can be called as autosomal gene, and those genes which control the sexual characters can be called as sex genes. Okay. The body characters or somatic characters or vegetative characters controlling genes are located within chromosome known as autosome, whereas the sexual characteristics controlling genes located within chromosome known as sex chromosomes or allosomes. You know that in human being there are two sex chromosomes. They are X and Y. For male it is XY in condition. For female it is XX in condition. Okay. Maleness is controlled by XY. Femaleness is controlled by XX. Okay, now you know that the body characters or somatic characters or vegetative characters controlling genes are usually located in autosomes, isn't it? But when a body character controlling gene is located within sex chromosome, such a type of character can be said to be what? Sex-linked characters. Okay, when that body character controlling gene is located within the X chromosome, it is known as X-linked gene, and such characters are said to be X-linked characters. If it is in Y chromosome, it is known as Y-linked genes, and characters are known as Y-linked character. These Y-linked genes are also known as Hollandric genes. Okay, remember Hollandric genes. And Y-linked characters are also known as what? Hollandric characters. For example, hypertrichosis, that is hairy pinna, presence of hairs on the outermost area, that is a pinna region. It is usually found in males. That's why it is said to be Y-linked. Okay. And the sex-linked characters can be classified into two types, and they are sex-influenced traits as well as sex-limited traits. Sex-influenced traits are those traits which is influenced by or affected by the sex itself and not by the condition of gene, whether it is homozygous or heterozygous. For example, baldness in human. You know that this baldness is commonly present in the male. Okay, so it is mainly depending on the sex of the organism and not by the condition of the gene. Okay, such a type of traits mainly influenced by the sex can be called as sex-influenced trait. Okay, then what you mean by sex-limited traits? These are characters only expressed in one sex. They may be caused by gene on either autosomal or sex chromosome. Example, female sterility in Drosophila. So my point is that in sex-influenced traits, the genes may be present in both male and female, but expressed only in one sex, isn't it? But in sex-limited traits, the genes are present in only one sex and expressed in one sex. That is the main point. Let's look at the option once again. Option A, Lesch-Nyhan syndrome. I already discussed this syndrome in the previous discussion. Lesch-Nyhan syndrome is characterized by the accumulation of uric acid, and those patients are exhibiting what? Self-mutilation. They are harming themselves. Okay, that's a unique feature of this syndrome. It is X-linked recessive disorder. You know that color blindness as well as hemophilia are also X-linked recessive disorder. Okay, and option A, option B, and option C exhibit crisscross pattern. So the correct answer for this question is option D, all the above. The next question is the DNA polymerase localized to mitochondria and is responsible for replication of mitochondrial DNA in eukaryotes is A. Alpha, B. Epsilon, C. Delta, and the last option D is gamma. You know that the DNA polymerase enzyme is the key enzyme in DNA replication. It was first discovered by a famous scientist namely Arthur Kornberg. Hence, DNA polymerase enzyme is also known as Kornberg enzyme. The actual DNA polymerase enzyme discovered by him was the DNA polymerase 1. Okay, and he got the Nobel Prize for this great discovery. Okay, what is the function of DNA polymerase mainly? It adds deoxyribonucleotides. Okay, thereby causing the polymerization of nucleotide chain. Okay. In the case of eukaryotes, the DNA polymerase are of different types. First one is the DNA polymerase alpha. Okay, it has an associated primase activity capable of synthesizing short RNA primer. You know that short RNA primer means RNA strand having 10 nucleotide bases. Okay, then DNA polymerase alpha is the only enzyme that could be involved in the primer synthesis during the initiation at origin of replication. So they are mainly concerned with the primer synthesis. Alpha polymerase is also required during the elongation step for the priming of synthesis of Okazaki fragments on the lagging strand. Okay, so the main function of DNA polymerase alpha is to synthesize primer. Then It is also concerned with the priming of synthesis of Okazaki fragments on the lagging strand. Okay. Okay, after the activity of DNA polymerase alpha, you can see the activity of DNA polymerase delta as well as epsilon. Okay, what is the role of DNA polymerase delta? It is a multi-subunit polymerase and probably functions at the leading and lagging strands on the replication form. Their main function is mainly concentrated on lagging and leading strands of the replication board. Okay, then what is about DNA polymerase epsilon? It consists of 4 subunits and its precise role in chromosomal replication is unclear, but it is believed that their activity is restricted to the lagging strand, perhaps only in the maturation of Okazaki fragments. Okay, so the clear role is still not known, but it is believed that their functional area is mainly concentrated on the lagging strand, mainly for the maturation of Okazaki fragments. Okay, then what is about DNA polymerase gamma? DNA polymerase gamma is found in mitochondria and is required for replication and repair of the mitochondrial DNA. Okay, all other DNA polymerase, namely DNA polymerase alpha, DNA polymerase epsilon, DNA polymerase delta, they are mainly concerned within the nucleus. Their activity is concerned within the nucleus. But the case of polymerase gamma, DNA polymerase gamma, they are mainly found in mitochondria and mainly responsible for the replication and repair of mitochondrial DNA. So the DNA polymerase mainly present in mitochondria and responsible for mitochondrial DNA replication and repair is nothing but the option D, gamma DNA polymerase. So the correct option for this question is the option D, gamma DNA polymerase. The next question is, which statement is not true regarding oogenesis in mammals? Option A, oogenesis commences at embryonic stage of the individual. Option B, each primary oocyte gives rise to 4 ova. Option C, growth phase is very elaborate. And option D is 3 polar bodies are formed. So you have to find out the wrong statement from these options. You know that the oogenesis is the process of formation and maturation of ova. Okay, the ova is formed from cells of germinal epithelium lining of the ovary. Okay, larger germinal cells with a bigger nucleus are called primordial germ cells. Okay, involves production of haploid nucleus as well as acquisition of food reserves and preliminary organization of cytoplasm. Okay, so oogenesis is simply the production and maturation of ova. This is a diagrammatic representation of different steps in oogenesis. Okay, when you are analyzing this diagram, you can see that from the primary oocyte, secondary oocyte and polar body formed, isn't it? Okay, and the primary oocyte doesn't give rise to 4 ova. Okay, so now we can make a look at the options once again. The option A, oogenesis commences at embryonic stage of the individual. It is a correct statement. Then option B, each primary oocyte give rise to 4 ova. That's the wrong statement. From that diagram, you know that each primary oocyte give rise to secondary oocyte and polar body and not directly 4 ova. Okay. Then growth phase is very elaborate. Okay, growth phase is very elaborate and well developed area. Then 3 polar bodies are formed. Okay, that's also a true statement. So the correct answer for this question is option B, each primary oocyte gives rise to 4 ova. That is the actual wrong statement provided in the option. Okay. The next question is, central thermoreceptors are located in A, hypophysis, B, hypothalamus, C, infundibulum, and the last option is pineal body. Okay, what are the main functions of hypothalamus? To maintain the body temperature, thirst, then appetite, and sexual desire, like that. So in order to memorize all these functions of this hypothalamus, I have created a card, just like a poem, in Malayalam. Okay, വിശപ്പ് ദാഹം സുഖാനുഭൂതി ലൈംഗികമായുള്ള ആസക്തി ഊഷ്മാവിൻ നിലപാലനം. വിശപ്പ് ദാഹം സുഖാനുഭൂതി ലൈംഗികമായുള്ള ആസക്തി ഊഷ്മാവിൻ നിലപാലനം. These are the main functional role performed by hypothalamus. And there is no doubt about that the thermal centers, central temperature regulatory centers, are located on where? It is on hypothalamus. Okay, so central thermoreceptors are also located in where? Hypothalamus. Hence, the correct answer for this question is option B, hypothalamus. Let us move to the next question. The most accepted link between Krebs cycle and electron transport system is option A, electrical, option B, physico-osmotic, option C, chemical conformational, and the last option is chemi-osmotic. Okay, you know that the respiration which is taking place in the presence of oxygen can be called as aerobic respiration, and the aerobic respiration involves the following step. First one is the glycolysis, second step is the oxidative decarboxylation, third step is the Krebs cycle, and the last step is the terminal oxidation or oxidative phosphorylation. Okay. You know that the glycolysis is the partial breakdown of 6 carbon containing glucose into 2 molecules of 3 carbon pyruvic acid. So, the end product of glycolysis is 2 molecules of pyruvic acid. As soon as the 2 molecules of pyruvic acid formed in aerobic respiration, they enters the next step, oxidative decarboxylation. These 2 molecules of pyruvic acid undergoes oxidation as well as decarboxylation, that is the removal of carbon dioxide, to form a 2-carbon compound known as acetyl coenzyme A. This acetyl coenzyme A then enters the Krebs cycle. Okay, and in Krebs cycle, this acetyl coenzyme A get converted into carbon dioxide plus water. Okay, and during all these processes, we can see the production of high energy compound, namely NADH and FADH2. Okay, but the body cannot utilize this, or cell cannot utilize these NADH and FADH directly, so they have to convert into their respective ATP. Okay, one molecule of NADH will give rise to 3 ATP and one molecule of FADH2 will give rise to 2 molecules. I already said that point in the previous discussion. Okay, so in order to produce the ATP from high energy compounds such as NADH and FADH, this NADH and FADH2 enters what? Electron transport system. The process which is taking place in the electron transport system is known as terminal oxidation or oxidative phosphorylation. Why it is called terminal oxidation? Because it is the last step of aerobic respiration. Why it is called oxidative phosphorylation? Because in that particular process, the phosphorylation is coupled with oxidation. That is, the production of ATP is coupled with oxidation, hence the name oxidative phosphorylation. Okay, so when we are analyzing the aerobic respiration, the glycolysis takes place in cytoplasm or cytosol. Oxidative decarboxylation takes place in where? Mitochondrial matrix. And Krebs cycle takes place in where? Again, mitochondrial matrix. And the electron transport system located, and the terminal oxidation takes place in where? It is in the inner mitochondrial membrane, especially in a particular structure known as oxysomes or F0, F1 particles. Okay, so here the question is that the most accepted link between Krebs cycle and electron transport system. Okay, during Krebs cycle, you know that high energy compounds such as NADH and FADH2 are produced. So, in order to convert this NADH and FADH2 into their respective ATP, it has to enter where? Electron transport system. Okay, in electron transport system, you can see the mechanism of formation of ATP. That is 3 ATP from 1 molecule of NADH and 2 ATP from 1 molecule of FADH2. Okay, and the mechanism of formation of ATP production, or the mechanism of formation of ATP during respiration, also during photosynthesis, is well explained by a hypothesis known as chemi-osmotic hypothesis. This chemi-osmotic hypothesis was first discovered by, or first proposed by whom? Peter Mitchell, and he got Nobel Prize for this great work. So, the connecting link between Krebs cycle and electron transport system from the given option is chemiosmotic hypothesis, okay, which explains the mechanism of formation of ATP. To explain chemiosmotic hypothesis, we require the following compound: a semi-permeable membrane, a proton gradient, then a proton pump, and finally the enzyme ATP synthase, also known as complex 5. Okay, to produce ATP, what are the requirements? The proton gradient. What is the proton gradient? Difference in the concentration of H plus ion is known as proton gradient. Okay, and also require a semi-permeable membrane which allows the entry of certain selected molecule. Then what proton pump? In order to pump a proton from one region to another region, a pump is required. Usually the pump is F0, F1 particle or oxysomes. Then what is requiring? An enzyme ATP synthase or ATPase enzyme. It is also known as complex 5. Okay, what is the role of that enzyme? This enzyme catalyzes the addition of inorganic phosphate to ADP to form ATP. That is, inorganic phosphate joined with ADP to form ATP in the presence of enzyme ATP synthase or ATPase. In respiration, this ATP synthase or ATPase enzyme comes under the complex 5. Okay, so chemiosmotic hypothesis is a hypothesis put forward by Peter Mitchell to explain the mechanism of formation of ATP during respiration as well as during photosynthesis. Okay, for this, for the production of ATP, the following components are required. What are the components? A proton gradient, a semi-permeable membrane, a proton pump, then finally the enzyme ATP synthase. Okay, so the correct answer for this question is the option D, chemiosmotic. The question is, xeroderma pigmentosum is a disease due to option A, production of guanine-guanine dimers in DNA, option B, defective melanin metabolism, option C, defective DNA repair, and the last option is autoimmunity. Okay, what do you mean by xeroderma pigmentosum? Xeroderma pigmentosum is a rare genetic disorder that occurs worldwide in all races and ethnic groups. Okay, first described by Hebra and Kaposi in 1874. Okay, and the main symptoms of this disorder is higher photosensitivity and premature onset or arrival of all major type of skin cancer. So what are the two main characteristics of this disease? One is the photosensitivity, and the second one is the onset of major type of skin cancer. Okay, early in the stage. Okay, so make a look at the second part of this slide. It is characterized by inability of cell to repair damage caused by UV, leading to genetic instability and skin cancer. Okay, so the point is that this xeroderma pigmentosum is mainly due to the deficiency in the DNA damage repair system. Okay, let us look at the normal condition, that is without the disease xeroderma pigmentosum. What happens? Under normal condition, when light falls on skin tissue, it will induce thymine dimer formation. Okay, the thymine dimer formation is a DNA impairment, and such a type of DNA is incapable of undergoing what? Replication, normal replication process. So that thymine dimer has to be removed from that particular DNA. So the induction or the production of thymine dimer as a result of exposure of UV light is simply called as a DNA damage. So DNA damage has to be removed properly, then only that DNA will be able to undergo normal replication. Okay, this slide actually explains the repair system in order to remove the DNA damage, namely thymine dimer. Okay. Okay, from the diagram it is very clear that at first that the thymine dimer region is recognizing, then unwinding of DNA takes place, then cleavage of that thymine dimer portion with little amount of normal portion, then filling that removed area with new correct sequences. Okay, it is known as sealing. Okay, in such a way that thymine formation, thymine dimer formation become rectified. This is a normal repair mechanism which is taking place. Okay. Here we are actually removing the damaged portion of DNA in terms of nucleotide. Okay, we are excising that damaged nucleotide. Hence, this repair system or mechanism is known as nucleotide excision repair mechanism. Okay, how this normal repair mechanism become abnormal? Okay, when the UV light fall on skin, what happened? The pole H gene get activated and it will normally produce what DNA polymerase eta enzyme. Okay, and actually this DNA polymerase eta enzyme protects cells from UV rays by inducing proper DNA damage repair mechanism. Okay, when these polygene become not activated, it will cause the disease, namely or genetic disorder, namely what? Pseudoderma pigmentosum. Let us summarize the major causes of xeroderma pigmentosum. First one is it is an autosomal genetic disorder, you know that. Okay, the nucleotide excision repair enzymes are mutated. That's why this disease is causing. Okay, reduced or eliminated number of nucleotide excision repair enzyme. Then metastatic malignant melanoma, then squamous cell carcinoma. These are the major causes of xeroderma pigmentosum. What is metastatic malignant melanoma? Skin cancer that has spread to other places in your body. Okay, it's also called advanced melanoma. Okay, what is squamous cell carcinoma? It's a cancer caused by an uncontrolled growth of abnormal squamous cells. I hope now you people know the correct answer for this question. That is, xeroderma pigmentosum is due to defective DNA repair. So, the correct answer for this question is option C, defective DNA repair. Okay, don't forget to study this DNA repair mechanism, because as far as your examination is concerned, I think the questioners will give some importance to this DNA repair mechanism. So, study very well all the details regarding DNA repair mechanism. Okay, the next question is GFE for fluorescent microscopy is obtained from option A, jellyfish, option B, silverfish, option C, pufferfish, and the last option is Drosophila. Okay, here GFP means what? GFP means green fluorescent protein. The green fluorescence protein is a unique protein that emits green color in response to blue or ultraviolet light. Okay, and is commonly produced by a jellyfish, namely Echoria victoria. And this GFP, as it is a protein, it is made up of 238 amino acids. Okay, and the color emitting region of, that is the fluorescent emitting region of this protein is known as chromophore. Okay, so GFE for fluorescent microscopy is obtained from where? The option A, jellyfish. So the correct answer for this question is option A, jellyfish. Let's move to the next question. The question is immunoblotting is option A, western blotting, option B, eastern blotting, option C, southern blotting, and option D is northern blotting. Let us understand the definition of blotting. It is a visualization of specific DNA, RNA, and protein among many thousands of contaminating molecules, requires the convergence of number of techniques which are collectively termed blot transfer or blotting technique. Okay, so the blotting is a separation technique which enables for proper visualization of the molecule which are going to separate. Okay. The blotting techniques are of mainly three types, and they are southern blot, northern blot, and western blot. Southern blot is widely used to detect DNA, whereas the northern blot is widely used to detect RNA. And western blot is widely used to detect protein. Remember, western blot is widely used to detect protein. Okay. The Southern blotting technique was first discovered by scientists named E.M. Southern, hence the technique is known as Southern blot. Northern blot and Western blot are named so because of the term related to Southern blot, and they are named because of discovered by Northern and Western. That's wrong. Okay. That is simply we are giving a term related to a pronunciation related to Southern, that is Northern and Western. Okay. Immunoblotting is nothing but it's a type of Western blotting. Okay. Immunoblotting works on the principle of what? Antigen antibody reaction. Okay, the product obtained as a result of antigen antibody reaction gets separated through immunoblotting. Okay. So in immunoblotting, we are separating the protein. The product's named protein, isn't it? So it comes under the category Western blotting. So the correct answer for this question, that is immunoglobulin, is a type of Western blotting. So the correct answer for this question is option A. Okay, you know that the ELISA enzyme-linked immunosorbent assay is also a good example for immunoblotting, so it comes under the category Western blotting also. Next question is, which of the following is responsible for the proofreading ability of DNA polymerase 1? Option A, 5-3' exonuclease activity. Option B, 5-3' endonuclease activity. Option C, 3-5' endonuclease activity. And the last option is 3-5' exonuclease activity. What is proofreading? You know that the DNA polymerase is the key enzyme that builds DNA in cells. Okay, so the DNA polymerase is a key enzyme mainly responsible for the synthesis of DNA or DNA replication. Okay, during DNA replication, most DNA polymerases can check their work with each base that they add. They are doing their work in correct manner or not? That is known as proofreading mechanism. Okay. And the DNA synthesis takes place in the direction 5' to 3' direction. You know that the deoxyribonucleotides are added to the 3' group. Hence, the strand become elongated from 5' to 3' direction. But the proofreading mechanism mainly involves exonuclease activity, removal of nucleotide from the end, and it is from the direction 3' to 5'. Hence, it is known as 3' to 5' exonuclease activity. So, the correct answer for this question is option D, 3' 5' exonuclease activity. Remember, the DNA polymerase 1 is having a proofreading capacity. It is associated with what? 3' 5' exonuclease activity. It removes the nitrogen base from the end of the DNA from the direction 3' to 5' prime. Hence the name 3' 5' exonuclease activity. Okay. Next question is, approximately 90 to 95% of blood T cells are option A, alpha gamma T cells, option B, alpha delta T cells, option C, alpha alpha T cells, and option D, alpha beta T cells. When you are analyzing the option, you can see the alpha gamma T cells, alpha delta T cells, alpha alpha T cells, alpha beta T cells, like that. All these are types of T cells mainly based on the type of polypeptide chain found in the T cell receptors. So they are mainly classified on the basis of their T cells receptors. Okay. In alpha gamma T cells, you can see the receptors consisting of alpha polypeptide chain and gamma polypeptide chain, like that. Okay. So remember, the most T cells are alpha beta T cells with TCR composed of two glycoprotein chains called alpha and beta chain. Okay, then in contrast, gamma delta T cells have a T cell receptor, that is TCR, that is made up of one gamma chain and one delta chain, like that. So the correct answer for this question is option D, alpha beta T cells. Okay, remember here the T cells are classified on the basis of the glycoprotein or polypeptide chain found in their T cell receptors. Okay.